Hey guys, so I wanted to walk you guys through some of these genetic notes and try to clarify a few things to help you with some of your practice problems. So, Mendel was this monk who studied pea plants, and he got really, really lucky in that he picked pea plants because they happened to have all of these different traits that were really simple, dominant versus recessive. The further we get into this unit, the more you're going to realize this is actually super rare. But pea plants were really useful when Mendel got started. So the big idea of Mendel and what he did was he had what he called true bred parents. So he had this purple parent and this white parent that he knew if he fertilized the parents with themselves over and over and over again, the purple parent would always produce purple offspring and the white parent would always produce white offspring. So he ran this cross where he crossed a purple parent with a white parent and got all purple offspring. So that must say something about purple being dominant because the white got covered up. So then he took this offspring and crossed it with itself, another self-fertilization. And when he did that, he saw this white gene come back out. So the white gene isn't necessarily lost, but it is covered up in some way. So you had 3 purple parents and 1 white parent come out of the self-fertilization of this F1 generation. So he knew, okay, purple must be dominant, but the white doesn't get destroyed. So what does all of that mean? Well, this was really early on. And so he didn't truly understand the meaning of all of his finding. And a lot of times this happens in science, and we find out later how important those findings actually were. So what Mendel actually got started on was this idea of alleles. So alleles are the different forms of a gene. In this case, for plant color or for flower color, you have purple as an allele and you have white as an allele. It's the same version, it's the same gene, different versions of the same gene. We know those different versions get separated during meiosis when we produce eggs and sperm. That's a process we've already looked at. So what Mendel started working on was these ideas of genotypes and phenotypes. So you can have a genotype, which is this blue word at the top. A genotype is the actual genes, the genetics that make up your DNA. The phenotype is what you end up looking like. And there's 3 or 4 kind of different versions of combinations you can have in this simple Mendelian genetics. So remember, we're working with the very simplest form of genetics at this point. So you can be homozygous, and homozygous just means that the alleles are the same. If both alleles are dominant, that means you're homozygous dominant. So that means you carry purple and purple, because that ended up being the purple, the dominant trait was purple. If both alleles are recessive, then you're called homozygous recessive, and you're going to show whatever that recessive trait was. In this case, in pea color, it was white. So homozygous just means the same. If it's homozygous dominant, it means it's whatever the dominant trait is. Homozygous recessive means it's whatever the recessive trait is. If you are heterozygous, there's only one option. You have to have what we call a big letter and a little letter, meaning you carry a dominant trait and a recessive trait. In simple Mendelian genetics, you're always going to look like the dominant trait in this situation because the dominant will cover that recessive trait. So that is how you end up with your phenotype, which is your physical characteristics. So I want to skip forward here a little bit. This should be familiar to you, the law of independent assortment, as well as the law of segregation. So the idea that as we go through meiosis, these genes get separated, the different copies, different versions that we have get separated into the cells. So this should have been review for you guys. But what should have been new to you is this idea of Punnett squares, unless you've had health class already. Ms. Serber sometimes gets to this before I do. So I want to show you guys, I'm going to hop over onto GoFormative to do this. But I want to show you guys how to get through these practice problems. So in practice problem number 1, it says, in people, freckles are dominant. Use a Punnett square to determine combinations among a woman who's heterozygous for freckles and a man who's homozygous recessive. So what we know, if the woman is heterozygous, the only option is for her to have a big letter and a small letter. I'm going to use the letter F because it's for the freckle gene. You can use whatever letter you want as long as you don't use X's. X's we're going to reserve for when we look at what's called sex-linked genes. So never use X's or Y's typically when we're doing this, because we have a tendency to assume those are the sex chromosomes. So if our girl is homozygous or heterozygous, we know she's big F, little f. Then we know the man is homozygous recessive. So he has to have, homozygous means they're the same, and they must both be little letters. Then you run your cross. The capital letters should always go in the front, so I tend to drag my capital letters through first. So these two both will get a capital F, and then all of the other letters in the combination are going to be lowercase f's. So then it says to determine the genotype and phenotype ratios of the offspring. So you have 50% of your offspring are heterozygotes. They have big letter, little letter. And then 50% of your offspring are homozygous recessive. They have little letter, little letter. Half of your offspring are going to have freckles because those freckles are dominant. So if they have one copy of the gene, they're going to show them in simple Mendelian genetics. And then half of your offspring are going to have no freckles because they don't have any of the freckle genes. They've got all non-freckle genes. Let's get this erased. Okay, let's try the next one. So question 2 talks about albinism. So it says albinism is a recessive disorder in which the cells lack pigment, causing the deer to be white. Cross a homozygous dominant deer with an albino deer to determine what proportion of the offspring will be albino. So albino is recessive, meaning you're going to have 2 bad copies, 2 recessive copies to be albino. So a homozygous dominant deer, I'm going to use the letter B for this because most deer are brown, so the dominant gene is probably brown. A homozygous dominant deer is going to be big B, because it's going to have only brown genes. Homozygous meaning the same, dominant meaning capital letter. Your albino deer is going to be little b. The only way to be albino is to have 2 recessive copies. It's the only way you show a recessive disorder. When you run the cross, always put your capital letters first. So we fill those in. And then they're all going to get a little b. So in this particular cross, when we talk about determining genotypes and phenotypes, there's only one genotype in the offspring, and that's the heterozygote genotype, meaning big, little. They have different letters, hetero meaning different. And there's also only one phenotype, and that phenotype is brown. None of these deer will turn out to be albino because they don't have 2 copies of their small letter. All right, let's run the last one and get this erased. And then the last one says, in sunflowers, fuzzy leaves are dominant over smooth leaves. Cross a sunflower true bred for fuzzy leaves with a sunflower that is heterozygous for fuzzy leaves. So true bred means that it will only ever produce fuzzy leaves, and fuzzy leaves are dominant. So that means, I'm going to use F's again because fuzzy is dominant. That means that your true bred must be big F. And then your heterozygote must be big F, little f, because that's what it means to be a heterozygote. When you run this cross, you're going to end up with your top boxes being big F, and then your bottom boxes being big F, little f. So when we talk genotypes and phenotypes, you have 2 different genotypes. You have the homozygous dominant genotype up here in the top, homo meaning the same, dominant meaning capital letters. And then you have 2 heterozygotes, meaning their letters are different. They have a big and a little. But all 4 of them will have the same phenotype, physical characteristics, meaning they will all have fuzzy leaves. So that is how you run Punnett squares. After you watch this video, I'm going to have an assignment on Classroom for you to do that has a few more practice problems. I think it's going to be an Ed puzzle as long as I don't change my mind. Check on Classroom for some more work to do.